In this video, we are going to study about eukaryotic DNA replication. The site of origin of replication in eukaryotes, it is called as ARS, autonomously replicating sequence. Like in prokaryotes, the origin of replication is called as ORI. In eukaryotes, the origin of replication is ARS. This region is AT rich region at the euchromatin region. Euchromatin region is that region which codes for certain protein. Eukaryotic DNA is polyrepliconic. Polyrepliconic means that there are many replicons in the eukaryotic DNA. A replicon, it is a unit of replication. It means in a replicon, the replication will take place continuously. The conserved boxes or sequences which are present in eukaryotes, they are A, B1, B2 and B4. These are the conserved boxes or sequences which are present at the ARS. Okay, by which the initiation proteins, they will recognize the origin of replication. So these are these sequences. Now, first of all, the initiation of replication. So, there are three steps in the whole eukaryotic DNA replication process. First is initiation, second is elongation, and the last one is termination. So, first of all, initiation of replication. Step 1. So, initiation begins at the G1 phase of the DNA replication, and some proteins, they are recruited at the ARS sites. Some proteins, they get attached at the ARS sites. And what are these proteins which are recruited at the ARS sites? These are ORC, CDC6, CDT1 and MCM. These four proteins will together form the pre-initiation complex in the eukaryotes. So this is step 1, that is the formation of pre-initiation complex. In step 2, all these proteins ORC, CDC6, CDT1, they get dissociated from the initiation complex except for the MCM. So except for MCM, all the three proteins, they get dissociated because MCM have a function that we will discuss. So at S phase, this happens at S phase. The step 2 happens at S phase. Specific CDK and cyclins, they will cause phosphorylation of the pre-initiation complex, which will lead to the dissociation of all the factors from the ARS except MCM. So the phosphorylation of the pre-initiation complex will lead to the dissociation of these three factors and MCM will remain. So in step 3, MCM will perform its function. MCM, the full form is mini chromosomal maintenance. So MCM is basically eukaryotic helicase. And what is the function of helicase? To melt the DNA, to unwind it. So it basically melts ARS. ARS is the origin of replication. So it melts ARS to form the replication bubble. And the replication bubble is formed in step number 3. In step number 4, RPA, that is replication protein A, it will bind to the single strands and will inhibit their re-annealing. As we have studied the function of SSB protein in the prokaryotes, single strand binding protein. So the function of SSB protein in prokaryotes and RPA protein in the eukaryotes, it is the same. That is, they will bind to the single strands and they will inhibit the re-annealing of the strands so that they are not able to bind again. Step number 5 is that MCM will recruit GINS and also will recruit CDC45. Now the function of GINS, it is basically to enhance the helicase activity of the MCM, to enhance the helicase activity of the MCM. And what is the function of CDC45? It is that it will recruit the DNA polymerase alpha. Okay, and we'll study the function of DNA polymerase alpha in the elongation process. So this is the initiation step, in which first of all there is the formation of the pre-initiation complex. Then at S phase they will get dissociated and only MCM will remain. Then the MCM will perform the helicase activity and will form the replication bubble. Then RPA proteins will bind and will inhibit the reannealing. And then in the last step, MCM will recruit GINS and also CDC45. Now we come to the elongation process of the replication. So in the elongation process, there are three DNA polymerases that are mainly functioning. First is DNA polymerase alpha, DNA polymerase delta, DNA polymerase epsilon. So first step of the elongation, it is performed by DNA polymerase alpha. It will form the primers at the replication bubble. So it will form the primers at the replication bubble. And also the DNA polymerase alpha, it is an RNA polymerase. In step 2, what happens is at the 3' end of each primer, pCNA will load as a sliding clamp. As we also studied in the prokaryotic replication, that a sliding clamp is recruited so that it can easily uh, guide the DNA polymerases to perform the replication process. So pCNA is proliferating cell nuclear antigen and it will load as a sliding clamp. It will slide on the DNA strand as a ring and will guide the DNA polymerase. So step 2 is pCNA. It loads as a sliding clamp. In the step 3, what happens is the polymerization will occur in the 5' to 3' direction in both the lagging as well as the leading strands. Now, in the lagging strand, DNA polymerase delta will perform the polymerization activity, whereas in the leading strand, DNA polymerase epsilon will perform the polymerization activity. And as we know that polymerization always occurs in the 5' to 3' direction on both the strands. In the leading strand, Okazaki fragments are formed. In the lagging strand. In the lagging strand, the Okazaki fragments are formed, whereas in the leading strand, the polymerization process will occur continuously. So, the step 4. In step 4, what happens is the primers are removed. Why? Because the primers are made up of RNA. They are made up of RNA and they have to be removed from the DNA. And the gaps which are formed after the primers are removed, they are filled. So the process here involved is there are two proteins which are involved actually, FEN1 and RNase H. FEN1 is flap endonuclease 1 and RNase H is basically digests the RNA. So what is the function of FEN1, flap endonuclease 1? It will cleave the bond between the RNA primer and the DNA strand. Basically, it will separate the RNA primer from the DNA strand. It will separate it. And RNase H, it will remove that RNA region which is left after the bond breakage. It means that it will completely remove that RNA from that region where the replication is occurring. So these two proteins are involved in the primer removal. The gaps which are created by the FEN1 and RNase H activity, it is filled by the DNA polymerase delta, which also performs the function of polymerization of the lagging strand. Now the last step of the elongation process, it is that the nick which is left, the nick that is left between the different Okazaki fragments, they are sealed by the DNA ligase enzyme. So this is the last of the elongation step. So what are the steps? Again, the DNA polymerase alpha, it will form primers at the replication bubble, and on 3' end of each primer, pCNA is recruited as a sliding clamp. Then DNA polymerase delta will perform polymerization on the lagging strand, and DNA polymerase epsilon will perform the polymerization on the leading strand. Then the primers, which are RNA, they are removed and the gaps are filled with the help of FEN1 and RNase H. Now the last step, that was the nick which are left, they are sealed with the help of the DNA ligase enzyme. Now the last step of the replication process, it is the termination of replication. Now in eukaryotes, the termination of replication occurs at the ends. There are special ends which are called as telomeres. These ends are called as telomeres. And here the termination of the replication will occur. And there are special enzymes. What are these enzymes? Special enzymes, telomerases. These telomerases these are the special enzymes which are reverse transcriptase, means that they will use the cDNA or the RNA segment as a template to perform the replication at the telomere sequences. Okay, so these telomerases enzyme, which are the special set of enzymes, they will help in the replication at these telomere sites. And here the replication completes in the eukaryotes. Thank you so much for watching.